So we've been working for a few years on developing a technology that was addressing objectives established by the Gates Foundation during Grand Challenges Exploration number 6. And could we treat fecal wastes using no water, be able to work off the grid, a whole list of requirements. And we believe at the end of phase 1 that we were able to confirm that we could do that. We wrote a proposal and they said, don't do that, just prove you can kill helminths. So that was our objective for this past year. And we did some work in the lab at Oklahoma State University. We did some design of equipment by computational fluid dynamics. It's nice to know the answer before you actually build something on how it's going to work. And then we went down to Durban and UKZN to build another unit and test it with sludge at that location. So we're using the concept of viscous heating, an old technology. An example would be melting polymers for injection molding is probably the most common application. So we're not adding anything to it. We're passing the solid waste through a shear field and allowing it to generate its own heat directly. So we're not transferring heat across any kind of a boundary condition. We're generating it by the molecules themselves. And this test not only established our ability to kill helminths, but we also looked at some operating conditions. And just a little bit on the background is one of our objectives is to be able to put this technology in a location where we can interrupt the transmission mechanism for helminths from one location to another, whether that's at the point of use or whether it's on a truck for delivering sludge from a pickup location. So again, viscous heating generates its own heat by friction, not fiction. You think you edit your slides, always one that gets through. By molecular generation, the amount of heat generated is controlled by the fluid viscosity. So we want high viscosity for this technology to work. Water is bad for us. We would like to be able to separate that out during diversion or some other mechanism. We can get to very high operating temperatures by regulating system pressure. We've operated one in the lab up to 190 degrees C, but the rate of generation is controlled by the shear rate. So we're basically passing the solids through a space between a fixed outer core and a rotating, fixed outer shell and a rotating inner core. And we can operate very high, but in fact, our preliminary analysis with Asper-Sum in Oklahoma showed that we really didn't need to. So we designed our system in South Africa to focus around temperature range of 70 degrees C plus or minus about 15 C. We got VIP sludge from Ethequini and screened it down to 1.2 millimeters to go through our gap size. We generated a concentration of eggs per 10 gram sample of about 300 of it by mixing 2 sludges from different locales. And during that screening process, our viscosity tended to drop, so we added, to get it back up, some dehydrated potato flakes as needed to achieve that. So just to show you, we've got a motor that's driving a pump, and then it comes in off the side. This insulation was just there at the time we took the picture for the case. We probably can do more with that. Passes through this gap space in here, and then there's a separate little motor that runs the rotation device. This is a hopper that feeds, and then we regulated the outlet pressure to match the required temperature, and then the system is only a single fixed gap and a single feed viscosity. So there's a lot more that we could actually do in testing the range of variability here. So we up again 50 to 85, RPMs tested 1100 to 1400, flow rates tested 1 to 4 grams per second, so that's on the order of roughly 8 to about 14 kilograms per hour for those units. 20 gram samples were set aside, and Colleen Archer did all the testing for the helmets, and she wanted me to particularly mention this slide. And so if we're taking samples, they were divided into 2 parts, one for immediate examination, and where you could determine by inspection where you thought they were viable or non-viable. But then there are half the sample was incubated and examined again after about 56 or 60 days. So the difference between these comes in if you think you've got a fertilized egg or the appearance of a fertilized egg, then over here you actually can get some growth, but there may be some that look like a healthy egg and still don't grow and end up back over into this category, into the infertile or dead egg area. So when I show you some result plots, the first one at initial examination is going to have some points that are higher than later. There's a little map that goes on with this, and if you really want all of this, give me your card and write slides on it, and I'll email you this and you can take a look at it. But it's basically a chemical engineering analysis of a fairly first-order kinetic expression. And this is in the literature from another scientist. And then how the data looks. So these colored lines are not a data fit. They're that model on what's expected based on literature data accumulated by other people. And this is the first case by the pre-incubation inspection, and we see activity of these cells dropping down to near zero, above 70, 75, and we operated up to, in some cases, up to about 85. But when I show you the next slide, it'll be this middle section that will move down the plot, and those actually give a better count after the incubation time. So we then use this model to establish what we thought were reasonable operating conditions for this system based on the moisture content values and the minimum temperature required by that prediction for 3/9 deactivation. So we really want to operate up and through here somewhere to be sure that we're not letting many eggs escape the system. We also did some energy analysis on the thermal efficiency. The question I get asked more than anything is, what's the energy relationship here, how much we're putting in, what it does. As we start feeding, there's an initial decrease in viscosity. And so what we found is that at high flow rates, the residence time is very small, and we're operating in some cases with residence time about 1 second. And our efficiency is quite high there, and in fact, this can extrapolate up mathematically to about 60%, but we don't have any data there. We're just following that model. And the efficiency drops off after you get that initial heat generation, and the viscosity of the material drops. Then there's more time for heat transfer out, and since we've got a fixed volume, then we end up measuring some of the conditions after some heat loss, so our efficiency drops down. We could actually make this thing much smaller and achieve the same directly. On the other side of the energy, I did a quick calculation. I pulled out a sample because the question was asked a little differently. At about, for a single data point at 78 degrees C, the motor to run the RPM, the energy measured out of the wall was 0.17 kilowatts out of a 1.5 kilowatt motor, where the flow rate was 1 gram per second, or 3.6 kilograms per hour. That works out to about 170 kilojoules per kilogram, which is about 1% of food, and for sludge, about 3 to 5% of the energy content of the sludge itself. So also, once we heat this material up, then the flow increases. So it's actually easier to move when it's warmer than, so we actually save a little bit on the pumping side. But, so we think we've shown that we can kill Helmus above 70 degrees, it's 90%. So for operating purposes, we've been looking at 85. And for 3 years of data on 2 different machines, we've never seen an egg survive that 85C in our reactor in a sample yet. Doesn't mean it's not possible, we haven't seen it yet. And again, the drier it is for us, the better. As long as it still can flow, we want it pretty thick. So, thank you.
Thank you very much. I'd like to invite the second speaker, which is Loïc Desclés, PhD student from Faecal, Polytechnique, Fédérale de Rotterdam, in Switzerland.
Hi everyone. So before starting, I would like to point out that I'm going not to look at like treatment technologies, but more what is happening on virus like during the digestion or storage. So let's talk a bit about the background. So viruses are basically simple microorganisms. So they are composed of a genome surrounded by a protein coat called capsid. And at that point, I would like to point out that the genome of the virus are, unlike other living organisms that are mostly double-stranded DNA, they have like RNA genome and also single-stranded. So viruses are excreted in high concentration in feces. We know from the literature that they are regularly found among resistant pathogens, with obviously the helminths eggs. And also from the literature, we've seen large heterogeneity in inactivation. So on the other side, you have the waste. The waste which represent harsh conditions for virus survival, especially because of mildly alkaline pH. And, some biocidal compound like ammonia. So the aim of our study is the main goal is to investigate virus inactivation naturally occurring in human or animal waste during storage or digestion. And this study is divided in 3 parts. And the first one will focus on MS2 model-phage single-threaded RNA in laboratory solution because it's easier to control. In the second part, we compare different viruses also in laboratory conditions. And finally, in the third part we challenge the conclusion made in the first two parts in real matrices like urine or sludge. So the first part is the inactivation. So we first look at the inactivation mechanism. So usually, like MS2 showed a log linear decrease of the concentration with time, and from this we could calculate the K-ops. And so we did that in with different solution composition. And you can then plot this K-ops against the activity of some compound of this solution, and you see that this K-ops is increasing with this activity, and you can calculate the Kj. And this is what we came up with. So we realized that actually this Kj showed a log linear relationship with the pKa of the base that could be present in solution. Some of them are present in sludge or urine. So in parallel to that, we also look at the mechanism of inactivation with biomolecular methods and also some specific assay. And we observed that the main part of the virus that that was damaged was the genome by cleavage. So putting those two facts together, so we came up with this mechanism that explains inactivation. And basically this is the base here that you can see according to its pKa, so its strength, that basically deprotonates the ribose of the RNA, which lead to the instability of this RNA and finally to the cleavage of the genome of the virus. So from this, we build up a simple model to predict the K observed, which is based on the bases in solution. So basically that's the sum of the product of the activity of the bases in solution, which is dependent on the temperature, the pH, and the solution composition. And also this Kj that I presented you before, which is a function of base pKa and temperature. So here you can see on this graph, so on the x-axis you have the prediction, and on the y-axis you have the observation, so the measured K-ops. So we tested this model in different lab solutions with temperature from 4 to 35 degrees and pH between 7.5 and 9.5. And you can see that the model fall into this plus minus 20% prediction, so which is quite good. And this model is now available on the web, so free of access, with a user-friendly interface. So basically, you have to input your temperature, pH, and solution composition. And you can get the activity of ions in solution, the kinetic of MS2 inactivation, and eventually the contribution of each base to this inactivation. So the second task was the second part is to look at the difference between viruses. So we tested 7 different viruses in different solutions. Here I only show you the one at pH 8 and 35 degrees. So here on the x-axis you have the K observed again. So we tested this in different solutions with increasing the base activity. And we tested 7 viruses. Among them there is 3 viruses, so echovirus, reovirus, and human adenovirus, and 4 phages. And they all represent they have different genome types. So here in green you have the RNA virus, and include DNA virus, which are subdivided into single-stranded and double-stranded. And the main message of this graph is that basically the single-stranded RNA viruses are more sensitive to this base than the double-stranded and the DNA virus. So that's a summary of what I just told you. So it will mean that DNA and double-stranded conformation of RNA provide resistance to inactivation. But why? What could explain this behavior? So especially for DNA, we know that the sugar of the DNA doesn't have this hydroxyl group that the RNA has, and that make it sensitive to this base. And this could explain why the DNA viruses are more resistant to inactivation. concerning the double-stranded RNA virus, the explanation is a bit more complicated, but to make it simple, the double-stranded conformation doesn't allow the base to access the OH group. So then, last part, probably the most interesting for you. So we checked those conclusions in real matrices. So let's start with the virus comparison in real matrices at 35 degrees. So I basically pick up MS2, which is our sensitive model virus, and all the other DNA phage that were shown to be resistant. So on the y-axis, basically to make it clearer, I normalize the K of the virus to the K of MS2. So everything that is more than 1 is more sensitive, and everything that is less than 1 is more resistant at MS2. And you can see that first in that solution, that when you add ammonia, you get the DNA virus to be more resistant. Then what happened in urine? So the two phages are still resistant, but surprisingly we see an increase of sensitivity of human adenovirus. So it's currently under investigation in our lab. But we think it's maybe linked to a specific compound in urine. And finally, sludge. So we haven't tested adenovirus yet. But for the phages, again, we see a more resistant phage, T4 phage. But for phi, we see an increase of sensitivity that is also under investigation, but could be due to adsorption to the particle. So last but not least, we challenge the model I showed you before. So in septic tank sludge with 5% total solid, 35 degrees and pH around 8, and urine samples, temperature of 20 and 35 degrees and pH between 8 and 9. So the gray dots are the one I showed you before. And here, so in brown you see the sludge, and in yellow the urine. So again, the model is quite good at predicting the observed value, except that maybe for urine we get a slight overestimation of the inactivation. So finally, the home message. So along the fact that we kind of address some of the virus heterogeneity that is shown in studies. So we were able to build a model that is with which you can predict MS2 inactivation. So it might sound obvious for most of you, but in order to get a grid of inactivation, a virus inactivation, sorry, you need to maximize pH, concentration of bases, and temperature, and that can be done with like airtight containers, solar heating, or additives, or in case of fluorine to avoid dilution. And last but not least, for the one of you that make a pathogen reduction study, we advise to use SS single-stranded RNA phage as a model, like MS2, as a worst-case representative of ssRNA virus, which are like enterovirus, and to use double-stranded DNA phage, like T4, as worst-case representative of double-stranded RNA virus, like rotavirus, or double-stranded DNA virus, like adenovirus. And thanks for your attention, and I put the address of the website.
I love your slides, and to follow where you were in the presentation. I think you need to show me how to do that. The final presentation is not by Professor Yetan. So I think the presenter needs to introduce herself.
Hi everyone, my name is Hanoi. I am on behalf of our project team to present our study took place 2 years ago. Our study names assessment of the composition of Faecal sludge in Singapore composting toilet. As you know, from yesterday, some information about the Faecal sludge from pit latrine or septic tank in Vietnam. I just threw quickly about that. Composting latrine are common used in rural area in Vietnam, and you can see on the bottom you can see the single wall pit latrine. It's only constructed above the surface and constructed by brick and cement. And here you can see the compost heap on the field. The farmer they made that for growth, and in Vietnamese condition with tropical climate, high temperature in the year, and OH recommend the Faecal sludge in dry pit latrine need to be stored at least 6 months before applying to the crop. And for the second one, the WHO guideline indicate that human feces should be stored at least 1 or 2 years. And our another surveys, we conduct survey to the rural area in the Red River Delta, and 66% of respondents store feces under 6 months, and 32% of households using fresh feces directly, like in vegetable crop. And Mr. Tu mentioned about the farmer conservation of rice. They keep the latrine. They keep feces in the latrine around 1 year. In the vegetable crop, they apply directly. And to it depends on the user way, different user way. Our objective is to verify the user manual of the VMOH. Thus, to assess 6 months is enough time to reduce the pathogen to acceptable level for safe reuse. And the second objective is to assess the decomposition process, and pathogen take place in a single pit latrine with storage time is the factor. Now the third is determine the need and improve decomposition process taking place in pit latrine. And the last one is to verify the role of bioadditive in decomposition process. At the beginning, we intend to conduct study in the field, but in fact, the user of the pit latrine, the single pit latrine, the new PC always adding every day, and we cannot access the pathogen with the storage time, and that's why we collect the material from the single pit latrine and transfer it to the lab, and try to describe the one cell of pit latrine in the laboratory. And here we have the 3 cylinder to describe the cell of pit latrine. And series 1, we make a cylinder like the red cell latrine with 5 kg of upper layer and 5 kg of bottom layer, and make a cylinder to see the reduction of volume compost material. And the series 2, we mix all the urine diversion toilet material and non-urine diversion toilet material into one mixer well by mechanical mixer. And where mixed material is fed into the 5 cylinder, and other 5 cylinder contain the mixed well material, and adding bioadditive to see what happened about the decomposition process and pathogen diet. The bioadditive we use has community include lactobacillus for reducing odor and antibacteria for decomposition of protein and cellulose. And all of them can active in a high pH condition because the users in the always add ash and lime after you, and that's why the material from the pit has a quite high pH, and this is some parameter we are to choose to assess the decomposition process and pathogen diet. Pathogen include anhaeminate, E. coli. This is some result we get. Here you can see the higher chain into the separate cylinder equal the separate latrine, with the red one is the high chain of material in the non-separating urine separating cylinder, and the other one is the separate material cylinder. You can see the high chain in urine separation toilet sample is less than non-separator toilet sample, but it's not so much different. Maybe I think it's caused by water content inside the non-urine separation material. And you can see here on the left, the top graph concerning to the TS and PS on non-bioadditive, and DS on bioadditive cylinder. You can see in the blue line, you can see the high chain in the each cylinder. It has a little bit different between the non-bioadditive and bioadditive, and ratio or TS decreased were observed with 50% of reduction after 9 months. Why we choose 9 months? Because MOH highlight said that the material they need to be stored at least 6 months, and we choose 9 months for assessment. And the next, you can see the volume of mixed cylinder decreased around only 10%. And here you can see the total nitrogen and phosphorus and ammonium. Ammonium has reduced significantly in the old cylinder bioadditive and non-bioadditive, around 90%, and can be caused by pH, high pH condition, or evaporation of ammonium, or the transfer from ammonium to nitrogen. And the next, the phosphorus, over the decomposition period is no change. And you can see the second graph, the blue one is the harmonic change in the bioadditive cylinder, and the red one show the change of harmonic in the in bioadditive. And you can see here harmonic and pathogen was found in both types of sample after 9 months storage, and decrease of harmonic in sample with bioadditive, and was more than in sample without bioadditive. And based on the data, the result, we give some conclusion and recommendation, like limited decomposition in passive treatment, like no mixing, no EC2 and anaerobic condition, and unpleasant environment, like too much lime and ash was added, contribute to slow decomposition process to poor microbial community, and nitrogen reduction is very high due to the high pH condition. And the next conclusion is a pathogen diode cylinder took place slowly even after 9 months of storage, and doesn't meet standard set by NOH for safe use. And that means we should try to find some information to improve the decomposition process, and pathogen diode, like we giving aerobic condition to composting process, using suitable bioadditive, and having further treatment outside of the pit, and using other adding material instead of traditional like ash and lime. And I'm finishing my presentation. Thank you for your attention.
Thank you very much. I suggest that you take all the seats, and I would like to invite the other two speakers too. Just a quick raise of hands. Questions for the first presenter? Two. Questions for the second presentation? One. I think people already have lunch on their mind. Oh, two more. And then questions for the third presentation? Okay, good. The first presenter.
Hi, David Brothers from Emory University. Thanks for your presentation. Really important things related to the asterisk variability. Can you, I don't know if and I apologize if I missed this, but what was the initial viability of the asterisk in your samples, and then what was your percent recovery from the protocol, and actually what protocol did you use for the specific viability testing?
I didn't write that section of the paper, so I can't tell you the authors' names, but it's included in the information, so I can get that for you right after. There was a control test done at each opportunity, so if you were to look at that plots that I presented, the ones on the far left would be in the range of the control. They may not have been the specific control value, but there would have been those values measured in near the C over C naught of 1 to 1.1. So, but I can get you the reference information.
Thank you. Second question. Katja, go ahead.
Hi, I'm from Hanoi University. If I understood correctly, did you screen the stuff before you went through the extruder?
Yes, we screened. One of our challenges is the small gap size that we have available. So as we're looking at upgrading this to the next stage, we're looking at integrating it with some kind of a tramp screening device that would take out larger debris. We have operated it, the system also with samples from baboon feces that contained a lot of sand that was actually the size larger than our gap space, and it just ground into smaller dust and particles, and we were actually able to handle that. But something like pennies or bottle caps, things like that, need to be removed.
Thinking of cleaners and things like that that go through our digestive system nearly untouched.
Right. Anything larger than our gap or can be compressed down to that is going to have to be screened out.
We have a third question in the corner over there.
Hi, I'm Elizabeth. I work with the Sustainable Sanitation Alliance. I would like to know if you've done already some engineering to look at the real life, how many years are you away from really using that in practice, and how does it compare cost-wise with other methods to treat sludges, like lime stabilization or sludge drying beds or those less intensive processes?
Right. The next one we build, I hope, will be a commercial application for a particular size. I envision like a standalone unit at a fixed location as being like a ablution block size that's used by several hundred people as being sort of a commercial scale. The other one would be hooking to a pumping system, which would have to be designed significantly different. What the economics would be relative to other technologies, I can't tell you. I don't know.
Okay, we have one more question for you.
This is a good follow-up question. Karen Nelson from UC. Compared to other just thermal heating disinfection technologies, I'm wondering how yours compares. Because you're still using electricity to dry it, right?
Yes.
You have the electrical conversion efficiency and then the thermal efficiency. So you could just be heating sludge directly with electricity or with natural gas.
If any time that you would have to add some other heat source, there's going to be some heat transfer. If you were using like a hot gas burned from natural gas, you'd have to have direct contact to have the same efficiency as this. We're heating at the molecular level, bumping against each other. We're not transmitting heat across some boundary like a metal wall or something like that to get to the molecules. So you can operate that. Those units will be bigger. We're talking about something the size of my fist that we're doing calculations. We think it can handle the solid waste from 1,200 people a day. If I'm going to transfer heat from any other source, I expect it to be significantly larger. It may be more economical that way. I don't know the answer to that.
Are you thinking of having heat exchangers to use, like, reheat the entering sludge for the exiting sludge as well and increase the overall efficiency?
No, time we've considered using recycle or recovering some of the heat. We can, if we reduce the gap size, we can boil off water at the exit when we relieve pressure. So in one case, we can take what may be a lower viscosity material, flash off some of the water, collect that, and recycle that back in. That would have some heat to it. But there's no real need for us to preheat it at all. We're getting up to temperature down a 10 centimeter section of this, which is our smaller unit, in about the first centimeter. So that's essentially our preheater is just sticking in the reactor and letting it go.
Thank you very much. Questions for the second presenter?
From the Center for Global Safe Water at Hanoi University, and I was interested in your model because usually in environmental virology, when I see models of die-off, they tend to have a long tail, and it's not so much that initial slope, but the long tail that may be due to aggregation or to absorption of particles. So I was wondering if your model takes into account the tail in inactivation. And then my second question is, I was wondering about your choice of viruses and why you didn't include hepatitis A virus, because that's usually, and I think of hepatitis A and noroviruses, I know you can't culture noroviruses, but hepatitis A usually has a reputation for being a very tough virus, and so I was wondering why you didn't include that.
Okay, so for the first question, so no, the model doesn't take into account the tail. For most of our experiments, so the one that were made in laboratory solution and the one that were made in urine and sludge, we've seen like a linear decrease. So even though we went till the detection limit, so but maybe there is some aggregation at lower concentration. So I should say that I don't know that. And the second question, so we had to pick some viruses, so why not hepatitis A? So again, yeah, I know from the literature that it's a strong one. But I knew it like recently, so so when I started my PhD, I just planned to work with a few viruses, and like ecovirus, we had this virus in our lab, so that's the reason why I picked this guy. Yeah, it would be, I agree that it would be interesting to look at hepatitis A to see why it is so resistant.
Okay, thank you. Second question. God knows.
My name is Annika Nordin from the Swedish University of Agricultural Sciences, and I would like to thank you for your very summary presentation. And I want to ask first if I understood, it was a lot of information, that the inactivation rate per day and concentration base was highest for the base with the highest pK. So that meant that carbonates were more efficient per concentration to inactivate.
Yeah, that's right, but so carbonates are, so you have carbonates and it has a pK around 12, but then depending on the pH you have also bicarbonate, and this bicarbonate has like a lower pK. So when you are at pH around 9 or 8, you have mostly bicarbonate, which is less efficient at inactivate than carbonate. So.
Okay, thank you. And my second question is whether you have considered to enter the data from other person studies on viruses and proteins into this model, because that would be really interesting.
Yeah, yeah, exactly. I did that, and I will do that for my thesis. But so one problem is actually an issue with this, or to address with this model, is that most of the time, I mean, for my experiment, for the urine and sludge, I characterize all the compounds in it, most of them. because I could do that in my lab, but most of the people, they just measure like ammonium and maybe pH and temperature. So there are some parameters that are missing. So I've already did like a comparison, and it's kind of really it doesn't always follow the model. Sometimes it's surprising. And also the point is that it's only a model based on MS2, so I need data on MS2, and there is some, but not so many for the moment. But you will see the results. You will be able to see the results soon.
Is there time for a third question?
Yeah, it's a short one.
Yeah. How do you think this model MS2 phage and its behavior in this model relates well to like FRNA phages from in general that you can find in environmental samples?
Sorry, I didn't get the question.
Like if the behavior of the MS2, if it will, you assume it to behave for FRNA phages in general.
Yeah, you mean if I assume the same behavior for the RNA phage in general? So yes, and I also assume the somatic phages, which are mostly DNA phages, to be more resistant. And I think that's what we've seen in some studies.
Okay. Thank you very much. Questions for presenter number 3?
Thanks for your really interesting presentation with some interesting results. I was particularly interested on your bullet point on one of your slides said the bioadditives had reduced the volume of the sludge, but I couldn't quite make out what the magnitude of that volume from your slides was, from your figures. Could you just clarify the magnitude of the volume reduction?
Can you repeat slowly the last sentence?
So you said the volume of sludge had been reduced. How much had it been reduced by, a percentage value?
Do you mean how? I did not catch your question.
So before you started the experiment and after you started the experiment, how many centimeters reduction did you find as a percentage of the total?
Percentage of total around 10% reducing.
Thank you.
Good. Second question in the corner.
Yeah, I guess it's sort of related. Did you do any statistics on the data between the bioadditives and no bioadditives? So, for example, with the die-off, it seemed like there were no replications on that.
There is no significant reduction of pathogen like E. coli. You can see. I can show you later the graph about the E. coli reduction. There is no difference, not much difference between the bioadditive and non-bioadditive. Even E. coli can regrow at the end of the storage around after 8 or 9 months.
Okay, any more questions? Otherwise, I think it's time for lunch. And if we're lucky, we must really Thank you very much. Enjoy the lunch, and we'll see you after the break again.